All right, welcome back, class. This is the Digestive System lab. It'll be similar to the lab we would have done in person. So on page 129 of your lab book, it kind of takes you through an introductory material, which I'm going to go through right now. For this part of the lab, you just have to do Physio X, the exercise 8, and only activities 1, 3, and 4. So look for that activity. You can upload your lab report on the module in the assignment, and then you'll have a quiz, and also this lab report will be due on May 13th, and then we're almost done with the semester. So we're going to look at chemical digestion and where that occurs throughout the body. Specifically, we'll look at the chemical digestion of carbohydrates, proteins, and lipids, and where that occurs. A little bit about digestion in general. We say mechanical digestion is just the digestion that your body does to the food itself. So mechanical digestion includes chewing and swallowing in the mouth, peristaltic mixing and propulsion in the stomach and the esophagus, more mixing and propulsion in the small intestine, and then segmental mixing and propulsion in the large intestine. Chemical digestion then occurs when we break down the molecules into smaller pieces or smaller chemical components. So chemical digestion of carbohydrates and fats begins in kind of the mouth area. Then the chemical digestion in proteins and fats continues in the stomach, small intestine. Then we get to including nucleic acids, amino acids, and it shows where everything is absorbed as well. So we'll take a look at the chemical digestion, again, of carbohydrates specifically first. This begins in the mouth with an enzyme called salivary amylase. Salivary amylase helps to break down starch into maltose, which is a disaccharide, meaning there's 2 sugars attached to it. Maltose will eventually be hydrolyzed and broken down into glucose, which is a monosaccharide. Pancreatic amylase is an enzyme, amylase, that will be what the pancreas secretes as a part of its digestive enzymes, and it will continue digestion of starch in the small intestine, the duodenum, where the pancreatic duct will empty into the duodenum. So the chemical formula for the breakdown of carbohydrates, also known as starch, we get starch plus H2O or water. With the help of amylase, that enzyme, we'll break starch down into maltose, which is a disaccharide. And again, enzymes in the microvilli of the small intestine will hydrolyze or break down maltose into glucose, which is a monosaccharide. And again, glucose is what we want because that will be absorbed by epithelial cells and then transported into blood capillaries within the villi of the small intestine. The chemical digestion of proteins begins in both the stomach and the small intestine. Pepsin is the enzyme in the stomach that will break down proteins into peptides. And pancreatic proteases are enzymes within the small intestine that will break down proteins. Pancreatic enzymes and brush border enzymes within the small intestine on the microvilli will cleave the peptides into smaller amino acids, which again are the individual components of a protein, and then these amino acids, which will then be absorbed into the capillaries within the microvilli of the small intestine. Then these amino acids are eventually transported to the liver via the hepatic portal system. And the main job of the liver is to detox all of this filtering stuff that comes through it via the digestive system. So the liver will take care of and filter, detoxify all of these amino acids that come through. So the enzymes that digest proteins will be pepsin in the stomach and pancreatic proteases in the small intestine. And they'll break proteins down into peptides, and the peptides will eventually be broken down into amino acids with the pancreatic proteases. Then the chemical digestion of lipids or fats is a little more complex. Before we even start to kind of we chemically digest it with bile as well, but bile is extremely important in the chemical digestion of fats. Bile salts, which are in the bile themselves, emulsify fats. And emulsification is the process of surrounding each fat droplet and breaking the fat down into even smaller droplets. That process is really important because it's kind of the first step in breaking down the fat into smaller pieces. So that's called emulsification, and bile does that. It surrounds each fat droplet and breaks the fat down into smaller pieces. If you remember from anatomy, bile is secreted from the liver and stored in your gallbladder where it's concentrated, and then it'll be transported to the duodenum, the first part of the small intestine, via that bile duct. So once the bile is within the small intestine, the duodenum, it'll help with emulsification of the fat, which is breaking the fat down. Once we get those smaller fat droplets, in the small intestine, pancreatic lipase, which is secrete via the pancreatic duct, through the ampulla, which is the same opening where the bile duct comes into the duodenum. The pancreatic lipase will help break down the fat droplets into monoglycerides and fatty acids. One thing that's different about lipid digestion is that fats will bypass the liver altogether, and they get absorbed directly into what we call lacteals. If you remember from anatomy, lacteals were part of the lymphatic system. And the lymphatic lacteals will transport the fat directly to systemic circulation. So fat completely bypasses the liver. In lab, you guys will work through and talk about optimal temperature and pH of enzymes. In terms of temperature, the optimal temperature of enzymes is normal body temperature, 37 degrees. If you remember that some temperatures can increase enzymatic activity. You'll also remember from enzymatic activity that pH of the enzymes is incredibly important, and this is especially true in the digestive system because different parts of the digestive system have different pHs. If we're talking about the stomach is an incredibly acidic environment, so all of the enzymes that help with chemical digestion in the stomach in the gastric juice will function best at a lower pH, because around 1.5 to 2, and that's because the stomach itself is very acidic, so these enzymes can handle that acidity and actually function best at the lower acidity. However, conversely to that, proteases in the small intestine function best at pH 8, which is just over normal, so it's starting on the basic side of things. The basic small intestine, the pH of the small intestine has helped to counteract the acidity of the stomach. So these digestive enzymes in the small intestine will function best at a pH of 8. Good luck with your lab report, guys. We just have one more lab to go. Good job.